Can you see presentation?
Yes, perfect.
Okay, so fine. First of all, thank you to give me the opportunity to be here today. Let me start. I'm sorry. Okay. Let me start my presentation with a brief introduction about the mechanism of cell mechanosensing and mechanotransduction. As you probably know, cells sense the externally force, like, for example, the movement or gravity, but also sense the external internal active force, like, yeah, for example, induced by the changes in the cell contraction or shape. And they transduce this mechanical signal in a more simple language for cell, which is written in biochemical and metabolic words. We have a long list of force-bearing proteins, including, for example, taline, which is important in the coupling between integrins and actin of cytoskeleton, but also, for example, we have lamins, important for the maintenance of mechanical integrity of nucleus, alpha-catenin with in the cell adhesion, and also titin. Titin has a K rule in the mechanosensing, mechanotransduction in muscle cells. This generation and sensing of mechanical signal determine the cell and tissue behavior in heart and disease. And the heart, for example, responds to mechanical cues with remodeling processes, tissue remodeling processes. And in these processes, they play a key role, the connection between cardiomyocyte and fibroblast. However, when we have, for example, the pressure overload in the heart, we have a tissue remodeling and such response like hypertrophy. When we maintain this mechanical overload, we can have the shift of the homeostasis of the cardiovascular system toward maladaptive remodeling of the tissue. As I say before, titin play a key role in the mechanical sensing and transduction cardiomyocytes. Titin has an important role to the maintaining of passive stiffness in cardiomyocytes, and has this mechanically active region in the A band, which is a spring-like region in which we have changing in the in the conformation of this region. Yes, for example, during the contraction relaxation of myocytes, we have the uncoupling or recoupling of the this uncoiled region, or the unfolding and refolding of immunoglobulin domain of the I band. So essentially, these conformational changes can alter the binding affinity for interactor. So in general, for the force-bearing protein, we have an active sites, and also we have a cryptic sites, which is a response to changing in forces. So for example, at low forces, this cryptic sites is buried in the structure, and so the site is off. But during the stretch of the protein, we have the unfolding, so the conformational changes, and the so the high force induces the exposure of the cryptic sites, and which is result active. Now, several approaches have been implemented to examine the protein mechanics. Like, for example, we can use, for example, the traditional knockout or knockdown or the overexpression of protein in cell. But in this case, we can alter the mechanosensing part of the protein, but also no mechanical function part of the protein. So the question is how we can study the role of protein mechanics in physiology and also in vivo, so in living matter. Our proposal in our project, we want to start the implement of the tool in which we can analyze and modulate the mechanical property in living cells and animals. And we decide to design these two models using titin, which has mechanical property, and we call these two models M-LOF, so mechanical loss of function and mechanical gain of function. In the mechanical loss of function is based on cleavage of the here the cleavage of the first transducing protein by TEF protease. So we have essentially a sequence for the recognition of TEF protease, and this cleavage locks the sites in the state of. Whereas in the mechanical gain of function, we using is achieved by the spike catcher system, in which we have the clamping of the random coil region, which leads the high force experienced by the domain. So hence we have in this high force manner the site is exposure and is maintained to the active states. Thanks to our collaboration with Julio Fernandez of the Columbia University and Wolfgang Link in Münster, my collaborators, my colleagues developed this mice model, the halotag TEF titin mice model, in which we have the TEF site in titin, the I band of titin, and also in the cell cassette we have this protein, the halotag protein, which allow us the labeling of titin in vivo. So they found that the insertion of the cassette is well tolerated in mice, and thanks of the halotag, we can see the structure of the sarcomere because we follow the titin in vivo, and also they found that in this insertion is maintaining, not perturb the structure of sarcomere. The ex vivo and in vitro experiment demonstrate that TEF protease mediate the severing of the titin, in which we have the TEF site of titin. So thanks to this model, we can follow in the protein in vivo, and our idea is to use this model also to following the severing of titin in vivo. So essentially, we want to check the effect of the M-LOF and NGOF model in cardiomyocytes, but also during the differentiation of cardiomyocytes and in vivo model. So to do that, we need an inducible expression of our mechanical effector represented by TEF, of course, in the M-LOF model, and also the spike catcher for the MGOF model. So to do that, first of all, we isolated mouse embryonic stem cell from halotitin mice, and we demonstrated that we can differentiate this line in cardiomyocytes, and also we can follow, we can see the cardiomyocyte differentiated thanks to the halotag in titin. So our idea is to use this cell line to generate a cell line in which we can induce the expression. We have so the structure for the TEF protein, and we can induce the expression of TEF in this model. So first of all, we building a lentivirus vector, in which we have, of course, the inducible promoter and TEF linked to the cherry tag, so a fluorescence, red fluorescence protein. As you can see here, the transfection infection using this vector, this strategy work well in HEC293 because we can see the fluorescence, so the positive fluorescence for cherry, but we didn't see the protein in Western blot analysis, so we can move it on. We can move it on another construct in which we have the same promoter, so a lentivirus vector, but without the fluorescence tag. And as you can see here, after the induction, cell express TEV after transfection and also infection with the virus. So the system work well with this vector. But an alternative tools that we can use to express TEV is also the using of adenovirus vector to express the mechanical factors. So for that, we tested another vector in which we have the cherry TEV protein in the adenovirus serotype 9 vector. And this vector works well in the HEC293. So we decided to move on the neonatal cardiomyocytes. So we infected this model of neonatal cardiomyocytes in which we have the positive fluorescence, the green fluorescence for the cardiomyocytes. And so we are able to see the expression of these cardiomyocytes, as you can see with the arrow, so the expression of TEV in these cardiomyocytes. So we decided to infect also the neonatal cardiomyocytes derived from the halotitin mice, and we found that we have the expression of cherry TEV after almost 10 days after infection. So we can say that we are able to express TEV in this cells, and also we found that the cell after infection, so after TEV expression, still remain alive, and apparently we have the beating of this cardiomyocyte is maintaining. However, when we analyze the contractility of these cardiomyocytes, we found that we have increasing of duration of contraction after TEV expression. So we can say that of course we have our MLOF in a model, and this model works well, and I can say also that we start to say an altered cardiomyocyte contractility induced by cleavage of titin. In the other end, we also started with the building of our MGOF model, and we validate this model in vitro, in which we test our recombinant target represented by our random coil region of titin with these two hand with the spike tag protein, and the recombinant effector represented by a bifunctional spike catcher with halo. So as you can see in the SDS page electrophoresis, we have the shift of our target in presence of our effectors. So essentially, we have the sampling of our model, in our model. So we can conclude that, of course, we are on track. So there are preliminary results, but we can say that the halotag make possible the following the protein in vivo. And I guess we can say also that the severing of titin makes possible the alteration of its mechanical function. And also it's important to follow, for example, the serial titin in the differentiation of cardiomyocytes. We need an inducible expression. So in the next step, we want to produce another mice line in which we want to introduce the cherry tab in ROSA 26 locus. And also we want to check if using another serotype of adenovirus, we have a more efficient infection of cardiomyocyte, neonatal cardiomyocyte. And finally, about the MGOF, we want to, of course, start the genetic engineer to introduce the MGOF model in mice. Let me thank the molecular mechanics of cardiovascular system group at CNIC, and especially Diana, Carla, Angel, and Manuel of my group, the core facility, our funding, and of course, thank you all of you for the attention.
Thank you, Maria Rosaria, for the nice talk.